Hi, everyone. Welcome to this workshop. We're really excited to have you. Today, we're at Illumina really passionate about incorporating and increasing or speeding up the adoption of NGS when it comes to routine clinical oncology. And today we have a great talk from Carlo, Dr. Carlo Bifulco and Dr. Brian Piening. Dr. Bifulco is the Chief Medical Officer of Providence Genomics. He's the director of the Translational Molecular Pathology of the Earl Chillis Research Institute and Providence Cancer Institute. Brian Piening is the technical director of clinical genomics, the associate member of the Cancer Immunogenomics Laboratory at the Earl Chillis Research Institute, Providence Cancer Institute. They will both discuss the details and the impact of implementing HRD assay at their institution and the added value of combining CGP testing and HRD testing. Thank you, Carlo and Brian.
Yeah, thank you, Pierre, for the kind introduction. So that's us. These are our disclosures. And this is a little bit what we've been doing for some time. So we have been running CGP for several years now in the context of a clinical study. It's a clinical improvement study, RB approved, where we actually do things a little bit different from what conventionally are done. So we run the test at the time of diagnosis by the pathologist, rather than waiting for the oncologist to see the patient at a later stage when they have failed therapies and so forth. So the idea here is really to improve the delivery of genomics data sets and change the way they impact clinical care. Ideally, we would like to see oncologists having genomic data sets at their first meeting and driving the treatment decisions at their first meeting with a patient. So we think this is very impactful. We give you a little bit idea of what we've been doing volume-wise. So I was mentioned before, we've been in this for some time, and volumes are quite significant. I think we presented at ASCO some initial results of this. We'll have more coming out. There's clear evidence of an impact actually on outcomes of patients in terms of targeted therapies. And this is part of a broader strategy that we have, which goes beyond just genomics. We have a strategy which is based on big data. It includes imaging studies, pathology, multi-omics, spatial biology, and the center of all this is AI. So we'll have efforts focused on this, and trying to incorporate all these data sources from a research perspective, but also from with the hope of improving patient care. And here too, we have stuff coming up online with publications in the press as we speak. But one piece that was missing so far was really HRD. HRD has been a big player for several years now in patient care. And PARPi inhibitors, you will hear a name later in the presentation multiple times, are big players. And we really had no way to deliver that through our comprehensive genomic profiling. And so we're super happy to have now and excited to have now the opportunity to add this on our repertoire. And the way we're going to talk today, I'm going to give a little bit of background for a few people who are not familiar with HRD in the room. And then Brian is really going to dive deeper into the specifics of assay. So to give you a little bit of background, we have 3 billion of bases of nucleotides in our DNA. We actually have 6 billion from 2 copies of this per cell. And there's a lot of activity trying to make sure that the DNA stays intact. And unfortunately, the DNA is actually easily damaged by multiple agents, radiation, ROS, alkylating products, and so forth can cause, can break the molecule. And all these pathways are quite important and preserved in evolution. If you go back in evolution, they're present early on. So they're fundamental things. And I will not walk through all of them. For reasons of time, as you can imagine, but they're all incredibly important. The mismatch repair genes, for example, MSI high, you all know how impactful that is for clinical care, immuno-oncology, and so forth. But we're going to focus here at the beginning of this sequence of pathway of response to DNA damage. And the first one I want to highlight is the very first, which is single-strand breaks. those are repaired by something that's called base excision repair mechanism. There's a complex, and PARP1 is going to be one of the players of the complex, and just keep that PARP1 in your mind. And then the second complex I want to talk about, which is quite complex and large, is the one which has to do with double-strand breaks. And you can imagine double-strand breaks are more difficult to repair. They're of all the things that can happen to DNA, they're one of the most challenging ones. And so there are many genes involved in this, and this is the most complex of the pathways that you see on this slide, actually, in terms of complexity. And here I want to highlight BRCA1 and BRCA2 and PALP2, and mostly the BRCAs here at the very beginning. There's also another pathway which works as well for fixing double-stranded DNA breaks. It is suboptimal. It stands for non-homologous gene fixing, but the homologous one is really the one which is most reproducible and the preferred pathway whenever possible. So what happens when things go amok, when those genes are defective? When it happens, actually, we have genomic instability, which is, as you well know, a driver of tumor biology. And there are several tumor types which have alterations of this, and because of the breaks, they end up having a lot of variability in their genomes, and often those are very aggressive tumor types. If you think about triple-negative cancer, high-grade serous carcinoma, et cetera, and that makes sense, right? You have so much variability, it translates in genomic instability, and the genomic instability drives very aggressive phenotypes. Now, all this biology gives us an opportunity actually to take advantage of what I just showed you for therapeutic purposes. And that's what happens with PARP inhibitors. So the way PARP inhibitors actually work is by doing something that's called synthetic lethality. So basically you have normal cells which can have one pathway defective, but they still survive and do well. Tumor cells do the same. But when you have two pathways defective, the cells die. And so if you have a tumor which has a damage in the HRR genes, which translates in HRD, then you actually, when you introduce an additional damage through the PARP inhibitor, which basically does a single-strand break, you end up having two hits to the cells, and those two hits are basically translating lethality for the tumor. And so that enables selective tumor killing, which is our goal, right? Selective cytotoxicity. So that's the key point from a biologic perspective. And so then the problem becomes, how do we identify those alterations which are making the cells sensitive to PARP inhibitors? And there are different ways. One way is to go back to the genes. You remember we had a list of genes. We can look if those are mutated, if they are altered, they have breakpoints, they have frame shift, they're deleted, they can be methylated. So there are multiple mechanisms for doing this detection. But there's also a big pathway where we actually don't know what is causing their inefficiency. And so looking at genetic background is not always the best way to assess this. And the other way actually is to look at the effect, which is the HRD component, which is basically the signature of that instability I was describing before. And there are several scars which are associated with that genotype I was mentioned before. And we can, this is really a phenotype. You have a genotype, the genes altered, and you have a phenotype, which is the HRD phenotype that I'm describing here. And here are some of the scars that you can see. The classical one is loss of heterozygosity, LOH. Basically is a loss of one of the two alleles that you usually have. But you also have alterations which are very similar, which are concentrated at the telomeric end of the chromosomes. Those are regions which are more difficult to keep in check because they are full of repeats, as you all know. So there's actually a telomeric alleelic imbalance very often. And then you can also have really chaos at the chromosomal level, which is basically large-scale state transitions at the chromosomal levels. And so those are all scars associated with the HRR genes defects, and basically are the phenotype of HRD. And so you can assess this from a clinical perspective as a biomarker for potential PARP sensitivity. How does this translate in the clinical landscape and oncology perspective? It's quite frequent, actually. So 17% of the tumors approximately have evidence of HRR mutations across multiple cancer types. And I want to highlight here really the orange and gray areas in these bars, because those are the ones which are related to BRCA1 and BRCA2. And the reason why I'm highlighting them, I do think, is because these are genes that have been known for 30 years almost now. They've been driving biology for cancer for a long time. Therapeutic efforts have been focused on those for a long time. So those are the most mature ones. And you will see that ovarian cancer, breast cancer, pancreatic cancer, and prostate cancer have a high component of BRCA1 and BRCA2 mutations as a driver of the HRD phenotype. And when you look at this from a therapeutic perspective, you will see that actually ovarian cancer, breast cancer, prostate cancer, and pancreatic cancer are all players here from a therapeutic point of view. You can see here a list. They all have different biologies as drivers of HRD phenotype. Some of them have germline mutations as a preponderant cause. Some have somatic mutations as a preponderant cause. Some have more variability in terms of what is driving the HRD phenotype. But they all share the same signature. They're all sensitive potentially to PARP inhibition as a therapeutic mean. And the one which is most well-defined and more adopted in clinical practice is serous carcinoma. So serous carcinoma, high-grade carcinoma of the ovary, is routinely now treated with PARP inhibitors after cisplatin-based therapies. You can see the biology which is behind this. BRCA mutations, as we were mentioned before, play a big role, but also you have things like methylation of the promoter regions of these HRR genes, as well as alterations which are less well-defined. Again, making the point for why it is so important to actually look at the phenotype rather than the genotype, because the genotype can be very complex behind the scene. And I think this slide really shows you what is currently done in ovarian cancer. I think we could have said similar slides for other tumor types. Ovarian cancer is the one that's most well-defined at this point of time, as I was mentioning before. You can see here a list of drugs which are used in this setting. The setting is maintenance, frontline maintenance after a successful cisplatin-based chemotherapy. And they make a difference in terms of PFS. And they make a difference in terms of OS as well. So very impactful, used routinely all the time, and really requiring a powerful and precise HRD test for driving the proper clinical decision-making process for the patients. And with that, I'm happy to share the baton with Brian, who will talk a little bit more about the testing itself.
All right, thanks folks, and thanks Carlo. So I'm going to do a deeper dive into the assay validation, so the work that we did around bringing an HRD assay online. And so as Dr. Bifulco mentioned, there's many ways to detect HRD deficiency. Looking for a mutation in BRCA1 or 2 alone is not always sufficient, because we can find these genomic mutational signatures or genomic scars that are indicative of HRD deficiency, even in the absence of a known pathogenic mutation in a causal gene. Let's see here. Okay, and so there, HRD is still defined in a number of different ways, depending on which group you're talking to. But one that's emerged over the last 10 years with a pretty solid base of data is this idea of a genomic instability score. And so this is taking into account 3 different types of genomic alterations that can be detected. A lot of this early research was done using SNP arrays, but through spaced spike-in probes can be done easily via NGS as well. And so what I'm talking about is loss of heterozygosity detection, telomeric allelic imbalances, which is again indicative of loss of homologous recombination repair, and again, as Carlo mentioned, large-scale state transitions. And so all that can be detected if you have essentially a genome-wide survey across a tumor data set. And so Mills and colleagues had previously shown that while each of these individual kind of mutational signature assays do show correlation with HRD detection, when you combine these things, you actually get some bimodal separation where your BRCA1 and 2 mutations, let's go back. do actually segregate into a separate distribution. And so here's your LOH, with your, in white is your BRCA1 and 2 pathogenic patients, TAI and LST, and you establish these scores here. But in the combined score, you really see the patients segregate over here to the right. And so, you know, this previous research has established a cutoff of a genomic instability score of 42, which is now used in the Myriad My Choice HRD assay, for example. So what did we do in bringing HRD in-house and within an in-hospital clinical laboratory? And so I'm going to talk a little bit about what we call PROVseq HRD, which we're in the process of bringing online, building a cohort and testing the assay on this cohort of real-world patient samples collected from across the Providence Health Network. And again, so now that HRD testing is starting to be more commonplace, it's become much easier to find these samples within you know, your existing data or sample repositories. And so largely that comparator testing was with patients that had been tested previously with the Myriad assay. Okay, so we started off with a cohort of 48 patients. We've since grown that cohort since then. I'm going to talk about the first 48. This is a work in progress that had previous testing and additional material available, so tumor material available. 19 of the 48 were positive for HRD by that orthogonal test, and 7 of those had a pathogenic mutation in either BRCA1 or 2. And then on the right, you can see the breakdown of the various ovarian tumor subtypes that make up this cohort. We also included 2 reference standard samples, so synthetic samples made by commercial vendors that worked really well, again, in contributing to this validation cohort. Here's an example H&E slide looking at the pathology of one of these samples, and you know, not a pathologist, so I'm not going to be able to speak too thoroughly on this, but you know, this is kind of what these tissues look like. For the samples in the cohort, we didn't have any that yielded too little DNA or RNA from the tumor. Every one of them yielded sufficient material for performing this assay. And so as Carlo mentioned, so PROVseq, the standard PROVseq solid tumor test is a test we've been running for a number of years in the lab. It's a 523 gene DNA and RNA hybrid test. I'll go over for the workflow for that. And then this HRD add-on is additional content that can be included or excluded from the core PROVseq assay. And so we can offer, you know, patients can, or physicians can order the standard CGP, or they can order the CGP with HRD. The workflow is about a 3-day workflow. I'll go into a little more detail, step by step, what we're doing here. But again, so most of the first day is tissue extraction and quant. Second day is library prep, HYBE, and 3, pooling, enrichment, et cetera, and sequencing. And so again, so for day 1, so as I mentioned, all of these samples had sufficient material for sequencing. Our standard workflow in the process is the tumor specimens are first reviewed by a board-certified pathologist. We require typically a minimum of 20% tumor. We then perform, actually it says micro dissection, but it's really macro dissection of areas rich in tumor that were circled. And the input for the assay is 150 nanograms of DNA. And again, we were able to get that from each of these samples. Library prep. So library prep, so standard library prep, we do shearing on a Covaris, looking for 90 to 250 base fragments. And again, I neglected to mention, so for the upstream extraction, we do a QIAGEN all-prep DNA and RNA co-extraction. That's largely done on QIAcube instrumentation, so fairly automated. And so the DNA and RNA are both input into the assay. So the RNA is typically used for detecting fusions. And so again, so overnight HIBE with the ProvSeq DNA, and in the case of HRD, with the HRD probe mixes. Library quants, generally not super variable. All of the samples that we made libraries for yielded high quality results. And then, down to day 3, enrichment and sequencing. So again, so we do, upstream enrichment steps, pooling, and then sequencing of the library pools. And so for this particular set of experiments, this was done on, relatively smaller flow cells, NovaSeq SP or S1, but you could move this into much bigger flow cells if you had a higher patient volume. Depth of coverage, looking at the depth of coverage for the DNA, this is average per base coverage across the targets. Looks pretty good. We had, one somewhat lower outlier, but still, about 375x coverage, so fairly high coverage. What does our informatics workflow look like? So some of this is, a mix of vendor pipelines as well as a lot of stuff built in-house. So I'll just walk through briefly what we do here. So data come off the sequencer, basically BCLs, FastQs. They're then input into a Dragon on-prem Dragon server. There's cloud Dragon servers as well where we can run these. But essentially, we're running a vendor-supplied pipeline, which does include algorithms for HRD assessment. Data come off that. We do largely most of our work in the cloud, so that's quickly moved to Azure in our HIPAA certified environment. And then we have a bunch of parallel processes that kick off. We do a lot of our work in the Databricks environment. So filtering, kind of standard variant filtering, annotation, linking to public databases, as well as we add on some additional fusion calling and other things, and those are largely Cromwell workflows. All these data get kicked into this big review software. This is an in-house variant analysis tool, but very similar to commercial offerings or platforms many of you folks have built in-house. And then from those data, we have variant experts, usually PhD scientists and molecular pathologists that perform various stages of review, and then that goes for report generation and final review by a molecular pathologist and sign out. And so again, once those data are available, they get delivered to our EHR. Ours is Epic, and so we've recently integrated the Epic Genomics module, so we're working on getting these data available as discrete structured data elements within Epic that we can then trigger alerts and whatnot when a patient is HRD positive, for example. Just a quick look for those interested at how much data that come off, these pipelines. So it's about 16 gigabytes, so gigabytes, not gigabases, per library. That's largely made up of FastQ and BAM, and then there's all these, additional files that get created that are useful, annotated variants, data associated with the HRD scoring, et cetera. And so what did our data look like? So again, we ran a cohort of 48 samples, 19 positive, 29 negative. Looking at the truth table, it looked actually tremendous. Like all cases that were called HRD positive by the clinical assay were also deemed as positive by the ProvSeq HRD assay. And similar for the negative, we observed the same thing, where all HRD negative cases were similarly called negative by our assay. Looking at the GIS concordance, so the genomic instability concordance, and so This is plotting, the original report we pulled from the clinical chart, the GIS reported on the MyChoice report, and that calculated from the ProfSeq HRD. And you see like really tremendous correlation between the MyChoice and the ProfSeq HRD scores. And again, you see kind of around that, you see that bimodal distribution, or we see a gap around 42, or that clinical cutoff for GIS positive. So kind of the remarkable thing is this was really fast. This was done on an order of weeks. We still have some more samples to run and analyze to get to a full validation cohort, but the data we've run so far looks really promising, and it's been relatively straightforward to get this assay up and going within our group. And so kind of the general conclusions, we were able to build an assay that easily tacked on to our current CGP. And so it really gives us a lot of flexibility, to sequence additional patients in-house that, may have had to, get sent out for another assay. And concordance looks tremendous with, assays already available, out in the field. And we're encouraged about this and are, we're working right now into moving this forward into a clinical setting. So there's a bunch of people we want to thank. So largely the main folks that contributed to this study, I'd like to give a special shout out to John Welly, who's our senior development tech, who did a ton of the work at the bench. Ben Cosgrove, who's one of our senior bioinformaticians who did a lot of the pipeline development, as well as the various members of the Providence Genomics team, as well as a number of our researchers, and lots of help from collaborators at Illumina as well in terms of answering questions and really helping to get make it as easy as possible to bring a HRD assay in-house. And I think with that, we can take some questions.
Thank you. Thank you, Carlo and Brian. We can proceed to Q&A. So simply raise your hand and we'll get you a mic.
Thanks, that was really interesting. You probably covered this, but I just want to clarify. So with regard to the HRD, if you were doing an HRD and an ovarian sample, for example, you would spike in additional baits for the HRD, but otherwise, so it's not a separate library prep, in other words. Is that correct?
Correct, yeah.
All right, thank you.
Great talk. Especially with ovarian cancer, you have tumor purity and poly-D. Before calculating HRD, do you handle that, or do you do median normalization?
That's a good question. I don't know the exact approach we're taking in the pipeline. I'd have to coordinate with some of our technical team.
Another question. With regard to the bioinformatics, is that within your own, on your own server, or is that you have to go to a cloud-based server for that, with the specific for HRD I'm talking? And how transparent is that bioinformatics?
Yeah, do you want me to take that? Yeah.
Yeah, so you have the option actually to run it on your local servers or in the cloud. We have done both at different stages. In general, we move to the cloud when we have very high volumes for a test, because often the on-prem infrastructure is not keeping up with the needs of the lab. And in terms of transparency, I think the pipelines from Illumina are actually quite transparent. I think they are, I don't know if people are familiar with this, but they're basically Dockerized containers, and you can actually look into the container content. So they have Cromwell's engines and stuff like that, WDL scripts, which drive a bunch of processes. Many of the processes are actually open source processes. For example, the variant annotation is Nirvana, and you can find them on GitHub. So there's quite good transparency in terms of the pipeline itself. Overall, we have been very, we found the solutions very flexible in terms of different patterns of deployment, and very, very nice to work with, to be completely honest with you. Right, yeah?
Yeah, and I'll say, like, no genomic data leaves our system or our environment on-prem or cloud. It's all self-contained, and so the Illumina-based pipelines are all run in-house.
Yeah, so even when it's a cloud environment, is our own cloud environment, and so there is our own tenant. They're all very well protected from an InfoSec perspective, so we feel very confident about the security, and it's not an external vendor, so to speak, except that Azure or Google or Amazon is the infrastructure provider. I think things are fully allocated and protected from that point of view. One comment on that: Providence is fully cloud right now from a clinical point of view, so all our clinical data sets are in the cloud. Epic data sets are fully in the cloud, so we're a little bit ahead of the curve. I think some other healthcare systems are still transitioning into that pattern. It has obviously advantages from a scale perspective, et cetera.
Yeah, so I have a question about more biological question. You talk about different subtypes, and do you get different scoring scores for different subtypes? And is there bias between different scoring scores? We do see that. We have seen not in other sarcomas also. So that's why I'm asking, my question is, do you see that subtype difference?
No, not yet, but I mean, with the caveat that we're, not a group that had focused on HRD previously, so this was really our first foray into running these types of samples. So I just don't think we have enough data yet to speak on that.
Yeah, but to the point, I think we will see what you're describing. That's my expectation as we run this out across larger populations of patients, right? Yeah.
Hi, thank you. Great talk. Quick question on the technical aspect. Not every tumor sample we could get 150 nanogram. Did you dilute it down, and was there an LOD?
That is in our ongoing validation studies. Those experiments are planned, but we haven't done that dilution experiment. But yeah, I think it's very important to look at what the LOD is going to be.
Thank you. Dr. Bifulco, you mentioned that the CGP is now being done at the time of diagnosis. I'm curious, has that impacted insurance and payers? But that's one, and then maybe I'll follow up with the second one.
Yeah, no, it's a great question. So what we did in the past was we ran this under a clinical study protocol or clinical improvement protocol, which was fully funded internally. And so the curve, the graphs that I showed you before are a representation of that. And that setting is very easy to do as long as you have the funding, as you can imagine. And the outcomes and et cetera we have are derivative from that. We are now transitioning out of that into a model where we would like to have a delegation from the oncologist to the pathologist of the initiation of the order of the testing. And the framework I would like to design is a framework where you have actually the implicit delegation with an opt-out mechanism, so to make things efficient. This is still work in progress. We still need to, we have agreements at the cancer center locally, but we want to extend this as a system. It's still work in progress. But that's where we would like to go. If that model is implemented, then you will have documentation of the delegation, and then hopefully from insurance perspective, no impacts because the delegation is formalized. But it is all work in progress, to be honest with you. And I do feel, as an aside, I do feel that it's a pain point for patients in general, because my personal bias is that we have decoupled genomics from diagnostics. Somehow they're becoming two different events. If you think about cancer, cancer is a disease of the genome. I mean, what we showed today is an example of that, but I think we could have hundreds of examples of that. And somehow we have decided genomics is an add-on driven by oncology at a later point of time, which results in basically in patients being suboptimally tested. So I strongly feel that it's a dynamic that needs to change, and we hope we'll be able to change in the future.
Great, thank you. And then Dr. Piening, there was one of the samples that had a lower depth of coverage. Was there anything special about that sample in your cohort?
I don't think there was, per se. I'm looking at John in the audience, if he remembers specific. Yeah.
Thank you.
So the H&E over here, yeah, the H&E photo that you showed looks like it could have come from a excision specimen of a ovarian serous carcinoma. So I was wondering, with other cancer types that are indicated for HRD, particularly pancreatic and prostatic, more often than not, one gets core biopsies, and maybe out of 12 cores, let's say of a prostate, you may have 3 with adenocarcinoma. So how do you deal with biopsies of really these 2 and more types of cancers? And so that's level 1, and it comes back to the LOD question. The level 2 is, let's say you have a core and you have a diagnosis. Do you wait for the excision in order to do HRD, to have enough material? Thank you.
Yeah, the great questions. You're right. So ovarian cancers often are bulky tumors. They're super easy actually from a testing perspective because they give you ample material. While not the same is true for prostate cancer, for example, especially in an early diagnostic setting, which is usually what you get. You get a prostate course rather than the metastasis and so forth. I have to say that we have a lot of experience with CGP in that context. I'm happy to tell you that we go down a lot in terms of quantity, in terms of input. And we routinely run CGP, the same test, for prostate biopsies, for example, even if the material is very low. We do the same on lung biopsies, FNAs, and so forth. I have to compliment whoever built the test because they did a very good job with the hybrid capture probes, and the yields are very good. Having said that, we have not done the work yet with HRD, as you heard. So we still have to determine the limit of detection, and so we'll see where we go with that. But it's certainly, if things hold true and the performance is similar to what we have seen with CGP in general, they may not be as bad as we may be concerned about. Brian, anything you want to add?
I'll say, like, when we launched the CGP a number of years ago, we kept alive a small amplicon assay with the idea that CGP may be more challenging for smaller biopsies with more limited input material. And we ended up just retiring the amplicon assay because really we were getting the same QNS rates, like a sample, and we had such a high success rate on just doing the CGP that it didn't make sense to run the smaller assay as well. But again, for HRD, that's something we're going to see whether that translates.
Any other questions? All right. Well, with that, thank you very much, Carlo and Brian. That was really enlightening. Thanks everyone for attending the workshop. And there's still some lunch in the back and some drinks, so please do enjoy it.
That's cool. Thank you.